Good morning, everyone. Thank you for attending the gene talk. So I'm Kevin from the Northern Hospital. So I'm research scientist, lab head, and from the Department of Radiology. So today the talk I'm going to summarize the INBRE support project because my INBRE, the funding period end on April 30th. So I'd like to talk about what we have done and also for our team, the research focus. So today, the title of talk, the clinical translation Fluorine-18 labeled Tryptophan Radiotracer. So the talk today includes the four parts. First, I like to give some background information. The second, I talk about the clinical studies of F-18 labeled Tryptophan radiotracer in two animal models. In the third part, I will talk about the clinical production of this radiotracer in a GMP facility. So, and most recently, there's a report from one state university. They already report the first human study for the same radiotracer. I just give a brief introduction of the same radiotracer for the clinical trials, and then give the conclusion of the study. So our lab mainly focus on the molecular imaging. So molecular imaging is a type of medical imaging to visualize what's happening inside the body in the molecular and cellular level. So there are multiple imaging modalities, such as optical imaging. And we also heard about the clinical practice. We most often probably heard about MRI, ultrasound imaging, and CT imaging. And different imaging modality is complementary instead of competitive. And today I'm going to talk about PET imaging, the following the positive emission tomography. They just use the radioactive material to look at the functional imaging of the human body. We know that the MRI compared to MRI, CT, or ultrasound, that's we mainly focus on the structure imaging to look at the whole body, the structure information, but did not provide the molecular cellular level information. So the PET imaging, they just provide the functional information. That's a major difference. But different imaging modality have the, you can see that from the table, have the advantage and limitations. So our research always from starting from the clinical questions. So we have very close collaboration with the clinicians to ask to identify the clinical challenges. So our ultimate purpose is to the translational research. We always start from the preclinical research because our research interested focus on the medical imaging agent development. So we are very interested in identifying the new target for the different disorders and develop new imaging probes and also develop new animal models. If all the preclinical imaging results are very promising, we start the clinical translation of the imaging agent and the new probe and apply the new probe, work with the FDA for the clinical translation. At the same time, a different pathway is because we are in the pediatric children's hospital, so we also work with the long probes because we work with other institutions. This is MGH and Methodist. They already develop imaging agent, but they only apply to the adult population. So we can cite their paperwork document. We can use the same tracer, but apply to the pediatric population. That's another pathway for the clinical translation of radio probe in our institution. So this cartoon shows what's the PET imaging agent. Just as I mentioned, the PET is called positive emission tomography. So this cartoon shows the major component of the radiopharmaceutical, also called radiotracers, also sometimes called radio imaging and radioactive imaging agent. On the right side, just to give you a typical example, is the cancer cells. On the cancer cell surface, they typically overexpressed different protein and biomarkers. And left side is the component of radiopharmaceutical. They typically consist of three parts. The first, we can see the radioactive sign. We introduce different radioisotope for the imaging, such as F-18 with the short half-life. Also, they can introduce long half-life for the alpha, beta nuclei for the therapy. And right side is the target molecular. Typically, the target molecular, they typically have high binding affinity and selectivity toward the target overexpressed on the cancer cell surface. And the radioisotope and the target, they typically link by a linker. So we design the linker, use a different linker to make sure the radiotracer have the suitable pharmacokinetics and pharmacodynamics and also the facility, let's say for the brain imaging, we need to design a perfect linker to cross blood-brain barrier and also water solubility and the physiological pH environment. So that's some of the major work we are working. So one of the key components is regarding the disease. When we look at the different disease, brain disorder, tumors, and also let's say the neurodegenerative disease, one of the key tasks we need to identify the target we are interested in. So here shows a very typical example regarding target. Let's say for the receptor enzyme interaction, and the FDOPA, that's a FDA-approved radiotracer for the neurodegenerative disease, and also have FDG, that's glucose for the metabolism. We know that the tumor cells cancers have high utilization of the energy, so they need to utilize multiple glucose. The third one is angiogenesis, because there's multiple overgrowth of the tumor cells. So they use the short half-life O15 water to look at the blood vessels and look at the new vessels generated during the tumor growth, and also hypoxia, F18 label F-misol, because we know that tumor have the hypoxic environment, so we can give us some imaging agent to look at the hypoxia tumor environment, and also the proliferation. We know that the tumor cell have overgrowth, overexpression, so the nucleoside tracer is called F18 label FLT to look at the tumor DNA synthesis during the proliferation. So here, just give a very typical examples. So what we are looking at, the biomarker for the different diseases. So today, the focus is develop F18 labeled tryptophan radiotracer to look at tryptophan metabolism pathway. We know that the tryptophan is a natural amino acid. However, tryptophan cannot be synthesized by our human body, but it must be obtained from external source. So in our human body, 1% tryptophan is utilized for protein synthesis, and 4% is utilized for serotonin synthesis. So in our human body, majority of tryptophan is metabolized through the cannulin pathway. The cannulin pathway is major metabolized moderated by two key enzymes called ADO and TDO. So under normal physiological conditions, the tryptophan downstream metabolite is called the quinolinic acid, can convert to NAD. NAD is the key enzyme utilized by our living cells. However, under abnormal conditions, excessive accumulation of quinolinic acid can cause neurotoxic effect and inhibit neuroprotective T cell functions. That's the cause of many neurological disorders, such as neuro-oncology, Parkinson's disease. Therefore, imaging cannulin pathway can provide non-invasive information for cancer immunotherapy. So next, I just give you two examples for the INBRE support project. The first one, we use this tracer for the imaging of medulloblastoma. So medulloblastoma is the most malignant pediatric brain tumors. So first, we apply the F18 label tryptophan tracer to the medulloblastoma tumor. So from the top left, we can see that this tracer have high uptake in the brain tumors and a very low uptake in the control mouse, and the showing B, and also the tumor was double confirmed by the HE staining. And we also compare the F18 label tracer with the FDG. FDG, just I mentioned, the FDA approved, is most widely used radiopharmaceutical in the clinic. So when we look at the bottom left, we can see that both tracer, F18 label tryptophan tracer and FDG, have high brain uptake. However, when we look at the blue line, the background uptake, we can see that the F18 label tryptophan tracer have significantly lower background compared to FDG. We can see that FDG have early time point have high background uptake, because amino acid, most amino acid in air, natural amino acid in air configuration. So we also compare the imaging result from the L and D configuration. We can see that the natural L amino acid have much higher uptake than the D counterpart at different time points. That means that the amino acid also uptake by the transporter. The majority is from the L transporter. So second example is we use a tracer for the neurofibromatosis type 1 animal model. So first we do the genotyping to identify the mice with the gene mutation. that have high chance to develop neurofibromatosis tumors. So right picture shows the typical example, the mice develop tumor. Then we do the biodistribution study from the wild type mice, the left side, and the mutation mice, the right side. And we can see that from both group, wild type mice and treatment mutation mice, both groups have high pancreas uptake, highest uptake in the pancreas, and also high kidney and liver spleen uptake. However, we can see that the brain uptake was low. Well, brain uptake was low, and also the bone uptake was low. The brain uptake is low because this tracer just the first example is that means that that's good tracer for the neuro-oncology. The bone uptake is low, that means that the tracer is stable towards differentiation. Sometimes differentiation is a major consideration, is a major concern during the F18 liver tryptophan tracer, because F18, because the bone typically have high uptake for the free fluoride. If the tracer not stable, the free fluoride enter the bone will be interfere with the imaging analysis. So we compare the imaging dynamics between the FDG and F18 liver tryptophan tracer. We can see that the two tracers, the heart uptake is still very different. You can see the FDG have the continuous high increase uptake in the heart, and the F18 liver tryptophan is a high heart uptake early time point, but decrease slowly. And we can see that from table, when we use a muscle as a reference region, we can see that the two tracers, when we look at heart to muscle and brain to muscle ratio, we can see that F18 liver tryptophan tracer have a much lower ratio compared to the commercial available FDG. That means that this tracer, when we do the clinical trial, this means that this tracer have less radiation exposure to the normal organ and tissues. That's particularly very beneficial for the when we do the future clinical trial for the pediatric population to reduce radiation exposure. Here shows the two example of the tumor mice. We can see that the top picture we can show the two tumor mice, and the bottom is a representative, the time point of the imaging of the two tracers. The left one, we can see that from left and right, we can see both radio tracers have high tumor uptake. However, when we look at the brain, we can see that FDG have lung-specific global brain uptake, and the F18 liver tryptophan tracer have a very low brain uptake. That further confirms that both tracer is good tracer for tumor imaging, but however, F18 liver tryptophan tracer have a significantly low background uptake compared to the commercial FDG. So we also perform the HEC staining to correlate the biomarker expression with the imaging result. And you can see that the tumor have high expression of the different biomarker, IDO, TDO, and the aromatic hydrocarbon receptor. And in the brain region and muscle have relatively low expression of IDO and TDO. And in the muscle have relative high expression of the another biomarker, aromatic hydrocarbon receptor. That's consistent with the reference report. So with the promising preclinical research in hand, we initiate clinical translational tracer. So we have the GMP room in our hospital site. So we synthesize the tracer by one part, two-step strategy. And the radiopharmaceutical was synthesized with high yield, high chemical purity, and in a numeric value, E value, and also high molar activity. The dose concentration is appropriate for the clinical translation. So the bottom picture shows the control panel because we use the control panel in the automatic system. The A is the control panel of the module. So B shows the inside the system hot cell, just for used for produce or radiopharmaceutical. So we perform 4 consecutive validation runs and test all kinds of parameters, let's say such as the pH, the residual solvent, the half-life, the molar activity, the endotoxin level, and the sterility to make sure all meet the criteria to clinical use. So the FDA requirement is just to conduct 3 consecutive validation rounds. So we publish the paper, we conduct 4 consecutive validation rounds and meet all the criteria for the clinical use of the radiotracer. Before we work with the FDA to submit the IND document for the clinical translation, we have the pre-IND meeting with the FDA discuss the toxicity studies, dosing study, and the radiochemistry section and the clinical protocol. FDA waived the toxicity studies and regarding other three major components regarding dosing study and radiochemistry section and the clinical protocol. FDA accept the other plan. So we prepare the document, prepare the whole package, and submit the FDA. This tracer approved by FDA for first human research. So during our ongoing clinical trial, another group from Wayne State University, they just recently report exactly the same radiotracer. They also report the first human study in the cancer patient. But this study involved a relatively small site. It includes 6 patients from 37 to 70 years old, and they two different tumor types. But the key thing is you can see that the injected mass, the mass of radiotracer is less than 1 microgram. So when we work with the FDA, submit our document, the radiopharmaceutical, the radio dose, and radio mass is less than, very small, less than 5 microgram. So that's why we also, in fact, that's why the FDA waived the toxicity study. So when they produce the tracer and we produce the tracer, the carrier mass of the F18 liver tryptophan tracer is essentially very, very low, typically less than 5 microgram. So the major consideration is we need to look at the dosimetry study to look at radiation exposure to the other different organ tissues. So here shows one representative study in this paper. They two examples. A is a 64-year-old woman and another one 67-year-old woman. One is the glioma, one the second one in the brainstem tumors. So when we look at that, the tumor align nicely with the MRI result, and also they use MRI identify the region of interest. Different MRI regions, we can see the hotspot. The different regions show the different kinetics, and clearly the R1 shows the higher standard uptake value compared to the 2 different regions, and also the result is consistent with the MRI result. And also for another brainstem tumor, we can see that the tumor to cortex ratio for the 2 cases and the SUV ratio, one is 7 and 1.5. So we can clearly see that the radiotracer have very low uptake in the background area and have high uptake in the tumor, and also show very high tumor to background ratio. Just to mention, one of the key components for the clinical translation is regarding the dosimetry, because when we were FDA, we submit the dosimetry study data in animal studies, but during the clinical trial, we still need to demonstrate that the tracer is safe for human use. So in this paper, they also report the dosimetry in adult human and the 4 patients. And the conclusion of the study is that the bladder is the most reliable organ. That means that the bladder receives the most of the radiation exposure, followed by the kidney and livers. So the conclusion of the study is that the bladder, the effective dose is comparable with the most often used clinical tracer in the FDA-approved radiopharmaceutical FDG. So they have very similar dosimetry. So that's further demonstrate that the radiotracer is safe for the human studies. So regarding the INBRE 2-year INBRE support, so the conclusion is for our research. So we validate the F18 liver tracer in 2 animal models. One in the medulloblastoma tumor, one in the neurofibromatosis tumor model. Regarding the neurofibromatosis animal model, from biodistribution study, we can see that the tracer highest uptake in the pancreas. And when we submit the paper, and the reviewer mentioned that maybe that tracer also a good tracer for the pancreatic cancers. That's a good comment. We can think about that. And also the tracer have also high uptake in the kidney liver. And the tumor uptake compared with FDG, but have significantly lower uptake in the background area, particularly in the brain and also in the heart. And also we can see that from the biodistribution study and the dynamic imaging study, we can see that the tracer is stable towards differentiation. That's one of the key components we need to think about during the new radiotracer development. And we also have the pre-IND meeting with the FDA and submit the document and produce the tracer in GMP facility to make the tracer suitable for clinical investigation. And during our study, we can see that from one state university of the same tracer already applied to the human study. We can see that the dosimetry of the F18 labeled tryptophan tracer is comparable to the FDA-approved FDG. That means that the tracer is favorable, acceptable dosimetry for the adult population. So next day we are going to move clinical trial. We can think about how to calculate the dose, apply the adult population to calculate the dose for the pediatric population, and to other disease types such as epilepsy, such as neuropsychiatric disorders and autoimmune disorders. That's the indication we propose in R&D. So last, I'd like to thank all the team members to the support of the project. I also thank our NEMOS, the histochemistry core, and the Sun University, Pennsylvania, Denver State University. They provide the imaging analysis. So just I mentioned, I really appreciate the support for the pilot project from the CTR, INBRE, and also partial support by the R21 grant and then the NEMOS Foundation. Thank you very much. So I'm ready to take any questions. Thank you.